Thank you everyone for coming, and I'd like to especially thank Felipe Toledo and the other organizers for inviting me. It's a really exciting conference, and especially as an amphibian biologist, it's been really sort of exhilarating to see so much amphibian biology being done in Brazil. Reptiles are fine too, but really, honestly, who cares? Frogs, all the way. So I wanted to begin with just a few comments in general about myself, and related to the museums, and sort of help frame my talk. So in my career so far, I've been lucky to be a student or a postdoc or a curator of 4 of the 8 largest museums for herpetology in the United States, right? So most of my science and career is due to that involvement with museum collections. And one of the things that I've experienced in going to different museums is that all museums, small museums, big museums, US museums, other museums, all basically have the same problems. So most museums today are limited by space, right? That's a big one. They're limited by resources and people in order to help facilitate the research that many of you want to do. And that's a challenge that faces every single institution, regardless if you're the Smithsonian or you're a small university collection, everyone faces the same. So actually, just out of curiosity, if you are a curator or paid to be at a museum to work on the collections, could you raise your hand? Okay, great, perfect. If you use collections, and whether or not they are specimens, they're tissue samples, or they are points on a map for niche modeling or something, could you raise your hand? Awesome. So hopefully you get the point, right? That there's many more people that are actually using museum collections than are actually inside the museum helping you to do that, right? So this is constantly a problem. For those of you that are interested in the physical objects themselves, right? Like myself, we do a lot of anatomy in my lab, as you'll see. One challenge is, and I'll be the first to say, I have loan requests to me denied all the time. Requests to dissect specimens denied frequently. Have any of you ever had a loan request denied? Oh, very few. Oh, it happened to me this week, actually. So, and there's always reasons, right? It could be rare specimens, rare species, rare specimens of species. It could be type specimens, right? There's all sorts of reasons why someone may not loan something to you, or someone may not want you to dissect that unique individual, right? So these are challenges that face really all museums, and for science that relates to the objects, these aren't anything new, right? These are problems that have persisted in museums for 200 years at this point. Okay, that's my context. So actually, Miguel's talk was great before. Just like Miguel, part of my lab and the time that we spend doing science is spent in the field, right? We're out collecting somewhere in the world, and most of the time, sort of the fun part, right? Actually, even in Miguel's pictures, the fun part is this, right? Actually being out in the field. This is the part that most people think when you say you're doing field work, it's like this. But of course, if you spend a lot of, and for my lab, we spend a lot of time doing this. This is just examples of field expeditions. We work mostly in the tropical Central Africa. Most of the time, though, when we're in the field, we're doing this, right? We spend all day, every day, preparing specimens, associating individual specimens with tissue samples for DNA or for RNA or for samples with skin samples for parasites or whatever, right? So spend an enormous amount of investment in making these, right? And this is this end product of much of our research. All right. These things will come back to museum collections like this. So this is just a picture of the inside of our collection at the Florida Museum of Natural History. And it's a really exciting time, not only to be a herpetologist, but to be anybody that works with museum collections, because information about these collections is more accessible now than it's ever been, right? So when I was a graduate student, which was not very long ago, I had to write people and say, hey, do you have any of this species, right? I mean, and you wait for someone to reply. I mean, now we don't always have to do that. Many museum collections are online. So this is just from this week. Images of, this is just simply using the search portal idigbio. This is mostly museum collections in the United States, but not exclusively. So just as an example, right, there's 3 million, almost 3 million amphibians on this list, and we know that it's a gross underestimate of amphibian specimens. Reptiles are the same. And in truth, for organisms around the world, right now there's, actually I think this is a little out of date, there's about 115 million preserved specimens or records available through iDigBio representing the world's biodiversity. So access to the information about specimens is better than it's simply ever been, right? But however, access to the objects themselves is about the same as it's always been for about 200 years, right? If you want to see it, you have to get it on loan or you have to go there, and there's all sorts of reasons why either you can't travel there or they won't send it to you. So we have these sort of pervasive problems that the access to the data is more and more accessible. It's easier to obtain, but the access to the specimens is basically like the early 19th, or well, early 20th or late 19th centuries. So one of the things that we've spent a lot of time in my lab talking about and creating are what I'd call digital collection objects. And I'll spend the rest of the time talking about how we make them and the projects we have. But I'd really like to argue that these representations of physical specimens that are digital, whether or not it's from a CT scan or a two-dimensional picture or photogrammetry, that these objects, they really add a lot of value to these natural history museums. They increase access to the physical stuff that many of us spend so much time in the field working to collect. It also really enables discoveries across disciplines, including disciplines that would normally not interact with museum collections, right? So traditionally we think of taxonomists looking at the specimens, but now there's a wide range of questions in evolution and biology in general, ecology, that would want to look at the physical specimens to get traits, whether or not it's for function or interpreting sort of functional traits in an ecosystem. And also, not only for research, but also for education, having these digital objects really facilitates new experiences for inquiry, for learning from actual physical objects. So this is a famous frog, so you'll see this frog a lot in my talk. So this is an African pig-nosed frog. This tends to be a group of frogs that's relatively poorly known. It happens to come from our field research in Angola. So the idea of a digital specimen is to take these physical objects that are in jars and to use some sort of imaging approach to begin getting data from it that can be shared quickly. So this is just an example from the vertebrate skeletons, all right? So there's many biologists of vertebrates that want to look inside things, look at skeletons, take them apart, look at pieces of the skeleton, such as the skull, maybe take quantitative data, in this case showing the relative densities, but you can also get surface areas and thicknesses, volumes of these data that are quantitative characters that you might want to use in various forms of analysis. In addition, we can also visualize things that aren't there, right? Spaces inside, in this case showing just the inner ear. But of course, not everybody is interested in only skeletons, so for people that want to interact with physical specimens, they may be interested in soft tissues as well. So they may be the surfaces of the animal, so there was actually a nice talk related to surface areas of frogs at this conference. You may want to take the skin off and look at other internal organ systems, right? And physically take them apart, whether or not it's cardiovascular system or muscles or nerves or skeletons or any of those other organ systems. So what we use in our lab to do this, and what I'll be talking about for the rest of the talk, is taking these objects, there it goes, and using traditional CT scanning, right? This is computed tomography based on X-rays, which I'll talk about in a moment, to create these data sets that are essentially showing the densest part of a vertebrate animal, in this case, the skeleton, right? And from that, you can create all of these range of digital products that can be used for descriptive anatomy. They can be used in quantitative analyses of function. They can even be used in things like this, like having 3D prints of things, right? That actually, personally, as an anatomist, these are really useful for morphology, but we also use them in education and outreach as well. Using other approaches today that include, in this particular case, which is often called DICE CT, for diffusible iodine contrast enhanced CT. It's basically taking an old frog, soaking it in iodine, and from that we can visualize the soft tissue. So it makes the tissues dense to X-ray. And from that, we can get all of these other soft tissues of the animals for vertebrates, including, and this will be the first bit of this in the talk, in addition to simply the anatomy of the animal, you can see we begin to get other important natural history data, right? So in the bottom here, we have, these are eggs, right? We begin getting size of eggs, volumes of eggs, numbers of eggs. We also get things like parasites. So for a lot of really poorly known taxa that are in our collections, for which there is literally no natural history, yes, this is only one specimen, but it can begin to fill in and begin that conversation about the natural history of these poorly known animals. So let me just say a word quickly about CT scanning, just so you know how these data are created. So most CT scanners have an X-ray tube on the left. The X-rays are electrons, right? The CT machine makes electrons that hit what's called a target, and the yellow here are representing the X-rays. Remember that X-rays are photons, right? The photons are hitting a detector that's electronic and being converted back to electrons. So when we put a specimen, any specimen, the X-rays are attenuated, right? The way that the X-rays move through the specimen changes based on its density, and it casts a shadow here. So just here's our cartoon example. The specimen is on a rotating stage, so as it moves, right, it's casting two-dimensional images, one after another, on the back of the detector. So to see, what this looks like in a vertebrate animal, here's a lizard from southern Africa. On the left, you can see this is just a series of two-dimensional X-ray images, right? It's not three-dimensional data. Your brain is creating it in 3 dimensions, right? So essentially what computed tomography does is that translation that's happening in your brain watching the two-dimensional images construct a 3-dimensional volume, which is on the right. So once we create these data sets on the right, this allows us to really take these animals apart in any way we want. It's not only this one view, this X-ray view, it can be any view. So we create this series of grayscale digital images that represent a map of the density of volume, represents a map of density inside this 3-dimensional volume. And once we have that in hand, we can do all of these other things that I'll talk about. So just to show that, once we have this volume, we can virtually dissect it any way we want, right? We can do it sagittally, dorsally, right? And of course, since it's based on density, we can also do what's referred to as thresholding, so only see things of a specific density, and that helps us often very quickly realize and visualize a skeleton of these animals. So what can you do with these data? So this is Ed Stanley, who's really a driving force of these data worldwide, but especially at the museum where he's a faculty member. And this is him in real time, right? He's taking a data set and quickly he's creating, in this case, a skull of that African frog that I showed you before. Once we have that 3-dimensional file, we can then do more things with it. We train students in high school, in secondary school, in college, curators, to actually do this work, and with some basic tutorials, you can very quickly get many people up to speed for creating these type of 3-dimensional data in the lab. So, of course, these data are great for all sorts of things. So, for me, I'm particularly interested in global frog diversity, and many frogs are rare, right? They're uncommon in collections. This, of course, is not a frog. This is Treticoena, the Brazilian lungless Sicilian. Thanks to Ana Pradente, who's also talking right now, we were able to get a very nice scan of these animals that are rare in collections. To me, as someone interested in natural history, I really enjoy being able to visualize other parts of the biology of these animals. So these are acanthocephalan worms inside the liver of a dendrobatid frog. This is pretty common to turn up in our scans. This is actually a very interesting approach to looking at parasitology, because we get in situ animals inside the host, right? And we can actually volumetrically calculate how much of this animal by volume is parasite. So we can do some other quantitative analyses of pathogens and parasites inside the animal. You'll notice there's actually very few lizards in my talk. This is one for you. It's inside the frog, though. So this is, sometimes we get surprises, right? So this is a Stereocyclops, this is a microhylid. Microhylids we think about as eating ants and termites. We only scanned one in this case, and of course it has a gymnophthalmid lizard inside of it. So sometimes we get very important small bits of natural history data from these scans. And of course this happens all the time. So here's examples of, the top left is a frog eating a frog, top right is a frog eating a crab. The bottom are two of the worlds really competing for who's the smallest of frogs. So on the left, we have Paedophryne from New Guinea. On the right, we have a species of Brachycephalus. The Paedophryne has an ant that's essentially the size of its head. and the brachycephalus has a single collembolin that is effectively the size of its head, just to give you a sense of how small this is. But these are taxa that have almost no information about their natural history and what they eat. So if you're doing some sort of macroevolutionary study and thinking, okay, how do we code this taxon about diet? It's a zero, right? You know nothing about these taxa, but we're slowly beginning to get information from digital imaging like this. So this is just a few examples of this to drive the point home. This is work for a graduate student in my lab, Dan Palou. We've been working with Santiago Castroviejo, who's here, on marsupial frogs. And so we're using three-dimensional imaging here to get important data about the egg size, egg number, egg position inside the mothers while they're brooding for marsupial frogs. And on the far right, you can see a species, this is Gastrotheca cornuta, that actually has the skeletons of these developing embryos inside her back. We're also, for Gastrotheca, actually particularly interested in larval anatomy. If you know about Gastrotheca biology, there's been a focus on tadpole anatomy. And so this is tadpole anatomy based on soaking tadpole specimens in usually either iodine or phosphotungstic acid, with which we can visualize the soft tissues. And actually many of the posters, both yesterday and today, had a figure that effectively looked like this middle one. This is sort of classical tadpole anatomy, sort of started by Richard Wassersug, who did a lot of SEMs on the buccal cavity. We can actually recreate those data virtually without dissecting the tadpoles using this approach, which is pretty cool. And as, of course, also being able to get really quantitative estimates of surface area and things like that inside. We also use this in my lab for comparative muscle anatomy. So here's an example from various front-first burrowing frogs, things like Hemesis or Nasicopetrachus from the Western Ghats of India. We can very easily visualize that muscles for making comparisons about function. And I think also, probably many of you saw yesterday's talk by Roberta that was on geometric morphometrics of snakes. This is probably one of the most commonly done things with three-dimensional data sets on a large scale, is actually doing geometric morphometrics to test hypotheses relating form and function. In this case, actually shown here, of trying to understand the sort of morphospace of frogs that have these heavily built ossified skulls, is a project that we are submitting very soon. Okay, so hopefully I've kind of painted a picture of the value of these data for a range of questions in biology. And so now I'd like to spend most of the rest of the time talking specifically about the project that I oversee that's referred to as OVERT. So OVERT, sort of short for Open Vertebrate. This is a project funded by the US National Science Foundation. And the goal of this project was to generate CT scans for more than 20,000 vertebrate specimens. And we estimated, and just because of the way we are funded, we're basically constrained to collections in the United States. So we estimated, based on US collections, we could be able to image about 80% of all extant genera of vertebrates, and then be able to have soft tissue scans, the sort of contrast-enhanced scans for representatives of at least half of all families. And then basically to take the data and just give it away and put it online. So that's a lot of what we've been doing. I was really surprised when this was funded, because there was an awful lot of attention that it got very quickly in like month one of being funded. And the thing to me that was really kind of interesting and exciting was, you know, these online science magazines here at the bottom, you know, none of these care about frogs, not a single one of them. This is a project that effectively is about dead fish, right? We have thousands of fish to scan, but we have things like Digital Trends or TechCrunch that are interested in these data, in part because of the visualization, but also it just sort of speaks to the value of these digital objects for exciting a wide audience in the public about biodiversity. And I think is a real a win for museums to be able to engage with this type of audience on a bigger scale to excite them about biodiversity. So this is a project that involves 45 PIs, and actually more than that, because there's people that actually do the work on the project, like all the technicians and students that help with it. We have 18 funded institutions, 16 of which are museums, and we have 6 imaging centers, because of course not every museum has a CT scanner, so we spend a lot of time moving specimens around among museums. And if you look here, you'll probably recognize most of the major US museum collections, especially related to herpetology. There's two exceptions. One is the Smithsonian, and we cannot actually fund the Smithsonian with federal money. So we have other ways of actually working with the Smithsonian, which we're doing right now. And the other is the American Museum, and just for whatever reason is not participating. All right, so this is our first sort of big project team meeting. You may recognize some faces here. Dan Roboski, who talked last night, is also a PI on this. But just to show you that we really have a diverse representation of people involved in museums across the US that have sort of agreed to make this happen. Not only for amphibians and reptiles, but for all these other major vertebrate groups as well. In addition to the CT scanning project, we have some partner grants. This is just a funding mechanism. So while they don't relate to amphibians and reptiles per se, I just wanted to tell you about them. So the first is a project that uses surface scanning. Essentially, you can think of it as photogrammetry or laser-based scanning of individual bones of animals that are too big to fit in a CT scanner. Whales, hippos, rhinos, things like that. This is led by Leif Tapanilla at Idaho State. So he has been spending a lot of time recently at the University of California at Berkeley, scanning individual bones of hippos and rhinos, and then virtually reassembling them so you can see an entire skeleton at once in a digital space. We actually have two other grants that were just recommended for funding, one of which is to take many of the data sets that are in the popular online sort of platform Digimorph, which is associated with the University of Texas at Austin, and actually usher those data sets into Morphosource so that they're finally published and available. Many of those data sets, if you work with CT data, you realize that what is available are pictures of data, not data. So we are now being able to make those data sets available, and many of those will be fossils, including many fossil amphibians and reptiles. We also have another partner grant that is focused specifically on rodent diversity that's coming. So the vision for this project was to basically do one of everything, right? One of each genus. To the extent possible, we are also spending our time doing sampling within species. So we are beginning, especially right now, of doing more sampling to do ontogenetic series, especially ontogenetic series, and capturing some variation within species that's of interest to the community. We can't do it for every species, but we can do it for key species of interest. When possible, we're scanning additional species within a genus, but another part that we're doing that I hopefully will resonate here is really building the architecture online to help anyone share their data, not just us, but anyone. So many people have data sets sitting on computers. The undergraduate or the graduate student that collected those data is long gone. You still have the data. And they're just sitting there, and you would like to make them available, but it's difficult. So this is actually a theme that came up yesterday in, this morning in Simone Denna's talk about the difficulty of sharing data. And part of it is that people don't know how to share data, and part of it is they feel like it's very time-consuming. So one investment from our project is building that infrastructure to make it, still takes time, but to make it less time-consuming and easier for you to do. And so just for reference, for vertebrates, there's about 10,000 genera and about, say, 1,000 or 1,100 families. So that's sort of the effort that we're trying to capture. Now, one of the things that's valuable, just to sort of explain how it works, is that we're really trying to build on investments that museums have already made in digitization. So first of all, when we go through, in the US, there were many projects like FishNet and Manus and Ornis and HerpNet, most important to this audience, that digitize those collection data, right? And that digitization just simply meant getting the label data effectively from the specimen into some sort of online digital platform. So we use that to discover what museums have specimens, but we also use those data to prioritize particular institutions. So just as an example, some of you may know of the Macaulay Sound Library at Cornell. Through Macaulay now, you can actually, it's a little hard to see, but you can actually sort all the media files in Macaulay by whether or not it corresponds to a voucher specimen. So we've actually been working with the Macaulay data to select specimens that actually have not only locality, but also have behavioral phenotypes, calls, videos captured of behaviors, to prioritize those individuals for scanning so that we're able to take an anatomical phenotype and relate it to a behavioral phenotype when possible. But of course, for the most part, we don't have those sort of complex behavioral data. But we can prioritize individual specimens that have other resources. And the main one that I care about is genetic resources. And so what we're able to do from the digital data that collections publish is to prioritize those institutions' collections of species that have associated genetic resources, right? So this is kind of a weird picture. This is just our internal operation of a Google spreadsheet. But this column is important to me because those are the institutions that actually have genetic resources, right? So if we're going to CT scan something, it would be great to know that it has a locality, that you can put it on a map, and then it has a tissue sample. Because if we add an anatomical data set to that, it's just going to make the impact of that data so much more that it's associated with a genetic sample and a locality, right? So we're trying not to scan things randomly. We're actually trying to put some effort into thinking about the things that we scan. Oh, and I should also just add that how we pick a species is important. Sometimes, we have many species to choose from, right? Many vertebrate genera are very diverse. So in the sort of one strategy that we've used is we use taxonomic databases to prioritize the type species of each genus. We don't prioritize, just to make it clear, type specimens. And in fact, I'm almost against prioritizing type specimens for many cases. And the simple reason that I'm really interested in capturing the phenotypic diversity of the species as represented by a single individual, right, which is complicated. However, there's plenty of examples where the type specimen, first of all, is damaged or is lost or is a juvenile, right? I mean, I always use Escaphus, the tailed frog from North America. It's called Escaphus because the female holotype didn't have a spade, right? It's probably the least interesting aspect of Escaphus biology, which is a frog that, of course, males have a penis, right? That's the only frog that has that. If we prioritize type specimens, we wouldn't capture that interesting bit of the phenotype of that species. So to date, we're almost finished with 2 years of a 4-year project. We've scanned more than 7,000 specimens across 42 US museums. These represent approaching 1/3 of all vertebrate diversity. Maybe it's not a surprise. Amphibians are much farther along than everybody else, but reptiles are right there behind them. Fishes, actually, by the numbers, we've scanned more fish than anything. It just turns out there's a lot of fish, so we're still working away on fishes. Amphibians will effectively be done this fall, where a representative of every amphibian genus should effectively be online this fall. And this is just a snapshot, which I'll talk more about Morphosource. So if you go into Morphosource and you browse by taxonomy, you'll see these numbers next to the taxon names. And so this is from this weekend. So as of now, there's 15,000 chordate specimens represented digitally in Morphosource, which is really astounding, right? There's an enormous amount of data. So far, not all of the data are yet up online, but we're working hard to have that happen. Right now, there's almost 5,000 specimens of those 7,000 scanned up online. And most of these are fairly high resolution. Most of these are scans that are visualizing anatomy between 20 and 50 microns. For a lot of fish and amphibians, it's actually even less than that. It's between, say, 10 and 20 microns. So this is very high resolution data. Most of these data sets, can approach 1 gigabyte per data set. So the way that we actually make these data available is through the online digital platform Morphosource. This is Doug Boyer, who's the PI for that. And so we put these data sets up online. They can be the three-dimensional mesh file, shown here, just sort of moving around a salamander skull, or they can be a zip file full of TIFF images, right? And you can download these, or you can use them, view them in the browsers that they have. Morphosource is getting very close to updating the viewer. So really, you can't view the CT data like you could on a standalone platform. Like if you downloaded these data, which can be expensive for the software, can be expensive for the computer. So this is what the new viewer will look like, where you'll be able to sort of digitally, in a web interface, be able to cut through. But more exciting than that is actually being able to render the volumetrics of that animal in a web viewer. So this is a platypus. We can actually change the density of it and begin to see its skeleton. This is not going to do all the things people want to do with CT data, but it's certainly more than people can do now using a web browser. So this will be a very big step forward for the website in the coming probably 6 months. So one of the things that I think is important as a curator, and I say this mostly from the point of view of years of working with NCBI and GenBank. which I've been frustrated by. So every time any of you type data into GenBank about your voucher specimens, you're probably repeating data entry that someone else already did, right? Someone has already typed that information, but yet you are retyping it. So it makes us very prone to errors, incomplete information. It's really, frankly, a waste of time. So one of the things that we've worked with Morphosource to develop, and is part of the effort to speed up your sharing of data, is using the API, which is basically being able to ping idigbio's information once you put in a number. So you can type in UF12345, send it in, and Morphosource will ask idigbio. Idigbio will come back and say, did you mean this one? And you say, yes. Then you just import the data into Morphosource. The nice part there is that we have this very structured data that comes directly from the museum collections now in the data depository. It also enables us to send data from idigbio, so we send the data back to Morphosource, and once we have that structured data in Morphosource, it enables to do things for the collections and to hopefully benefit the scientific collections, which I'll say more about in a sec. All of you have probably had the experience, or many of you, of uploading one gene sequence at a time to NCBI. Now you can upload many at a time. It's still a pain. It's similarly a pain for data sets like these, which are large files. So we've worked a lot on actually creating Python scripts to help you fill out batch uploading data sets to get them onto Morphosource quickly without spending tons of time copying and cutting and pasting and things like that into data sets. And so this is mostly developed by a woman who's actually starting a faculty position at Stony Brook this fall, Natasha. But here's the bit.ly link if you're interested in those scripts. It's actually very helpful. So once we have those data, it enables us to collate information by collection and give it back to the museums. So that if the Florida Museum of Natural History Herpetology collection manager wants to find the data for our collection, he doesn't have to wade through all the information for the Smithsonian and the Cal Academy and all that, right? So we can effectively give it back to museums that they can then integrate into their databases. And so anybody can see this, by the way, if you're interested in how things get used. Morphosource now has a data reporting page, which notably NCBI lacks. So you can actually get the data per collection, per institution, to learn how these objects are actually being used. And so that's actually how I am able to get this type of information. So in the last year and a half of working on OVERT, these media files have been viewed about 130,000 times and downloaded more than 5,000 times. And actually, it sort of grows almost exponentially by the week, the views and the downloads. And some of the most popular things on the site, unsurprisingly, are big things like primates. So we've CT scanned gorillas, chimpanzees, not whole crocodiles, but parts of crocodiles like this gharial here. And just to give you an example of how impactful it can be for a specific collection, so this is just from the collection I oversee at UF, we have only 400-something specimens up on Morphosource. They've been downloaded 3,600 times. I think we probably have the most looked at tuatara in the world at this point, just based on downloads and views. This is an example for many squamate systematists. They care about the crocodile lizard, Shinisaurus, which is a monotypic family from China. There's relatively few of these in scientific collections, so this is just the numbers on the bottom right about this. This thing, once we put it online, quickly started getting used by systematists, people coding characters for phylogenetic analysis all around the world. So individual data sets like this can have a big impact worldwide on, especially for research. So this is just an example of the types of things that people are actually using these data for, and I think really adds value to the scientific collections that they come from. And actually for this conference, I only pulled out faculty and graduate requests from Brazil. And there's more. These are just the ones that fit on the slide. So just to give you an idea of how diverse the research uses can be, you'll notice that it includes things like looking at ecomorphology, 3D geometric morphometrics, more or less sort of basic descriptive anatomy, comparisons with fossils, more comparative anatomy, systematics, and evolution. Another thing that's really important are the uses for teaching and other uses, right? So, many museum collections, pieces of them get used for things outside of research, right? There may be a specimen on exhibit. You may loan a specimen to a classroom for teaching. Morphosaurus actually has exactly that same impact, which is really interesting. So we've started interacting a lot with teachers, artists. Even on the bottom here, if you can't read it, it says 3D print for surgery planning. So people from zoos have been downloading data sets to plan the surgeries on animals at zoos. So it really is having a big impact. About half of all of the requests of these data are for research, but the other half of the downloads are for this other range of uses, especially for education. So we've really been interested in how to more effectively engage people doing education with these. So we have people that are using three-dimensional data, often through 3D prints, but not only. Often through three-dimensional prints of things like this. They're using them in classrooms for children. They're using them in teaching undergraduate anatomy. They're using them to teach art students what the inside of animals look like if they're trying to draw them or illustrate them or animate them. So we've been working actually directly with teachers because this has been an important thing we've discovered about the impact of these digital museum specimens. So in June, we led a week-long workshop with teachers in the state of Florida, developing lesson plans, examples of how they might use them in the classroom, whether or not it was a 3D print or a picture of the data or a digital 3D model. And so we spent, actually it was a very useful week for me to understand the limitations that teachers may have in the classroom, but it also really showed the enthusiasm that many people have for interacting with real museum objects, right? Just to draw that point again, you know, these are things that are just sitting in museums that now digitally there's kids in classrooms interacting with that specimen and all the story behind it. So one thing that we've been experimenting with is how to most effectively share these as digital models for education. So I mentioned this to someone earlier today, a Sketchfab. This is a free website. You can get the app on your phone. You can look at digital models on your phone. So when I taught herpetology this year, we actually created a collection, a digital collection for herpetology that students could actually interact with outside of class for learning names of bones and things like that. So the example here, just to show you, here's a Dermophis mexicanus specimen, a skeleton of a skull of a Sicilian. Many students, you know, wouldn't know the name, so we can label those things. And importantly, we can also embed images. So that really helps us have a pretty rich example for teaching students anatomy, whether or not it's herpetology or comparative anatomy or just a general natural history biodiversity course. And so I think this is a very powerful thing that any of you can do. It's free. If you have data like this, you can make your own collections and models like this and really have a big impact in teaching, especially since it's on phones and all of you have phones. So it's an opportunity for us to engage people that way. One of the other things I'm really interested in, you know, we can make 3D prints, which are great, but they're also not really how we interact with specimens, right? As scientists, we take things apart all the time, right? So it's an interesting experience to take things apart, for whether or not you're learning the anatomy and the structure of things, or you're just interested in how things work. So this is an example. This is Ramon Gonzalez, who's based in Spain, is an engineer who simply started posting YouTube videos of our data as a three-dimensional puzzle. And the reason he did this was because his wife is a secondary teacher and was teaching biology and wanted something more dynamic for teaching anatomy. And so we've spent some time corresponding with Ramon to understand exactly what he was doing. This is a very, very high-end, expensive version of a 3D puzzle. So I'm interested in things that are cheap. So I have two students in my lab. The ICVM meetings, the Vertebrate Morphology Congress is in Prague right now. So this is our lab's poster in Prague, one of the posters. It's all about making these puzzles on your own 3D printers. And effectively, if you have a cheap 3D printer, you have some digital files, some magnets, which you can order off Amazon for $15 maybe, and paint, you can make colorized three-dimensional puzzles for classrooms and things like that. So, you know, in a museum, sometimes many people in museums, including me, interact with the public, often interact with students. So being able to have these sort of more dynamic examples is great. And importantly, right, this is not ordered from Carolina Biological Supply or any of these other companies, right? This is a specimen from fieldwork that we did that has a story about why we're doing science. You know, this is a valuable opportunity to teach people about the science that we do in museums and in the field. I'll just say kind of in closing, hopefully we have time for questions, that if any of you are interested in learning to do this, there are opportunities actually funded through our grant. It doesn't matter, I've been told it doesn't matter, if you're a US citizen or not a US citizen. We have a sort of internal grant application process that you can basically suggest a week's worth of work, go to Friday Harbor at the University of Washington, hang out with Adam Summers, and CT scan of stuff for your own work, all right? And to learn those skills, both in terms of generating the data and working with the data, really only on the caveat that we basically post the data online then. But that's because that's the mandate of our project, but it's also an opportunity for you to generate 3D data and learn how to do this work yourself. So I just want to just close with a couple thoughts. So effectively what has happened in museums in the last, I don't know, 20 years has been what we've in a recent review article termed digitization 1.0, right? It was the transforming the physical stuff into some sort of digital representation, right? It could be just simply the text that describes the species and the place and the date and all that. Or it could be more sophisticated things such as, you know, taking a two-dimensional image of it or creating a three-dimensional model. That's really the first version of digitization, because some of the most exciting parts about this are coming next, right? So once we have that in hand, we have workflows to where we can add new layers of data to these digital objects, right? We can annotate them with characters. We can add other types of layers of information to those digital objects, sometimes using fully automated approaches through machine learning now. So it's a really interesting time to begin thinking about what can you do with these large-scale data sets representing biodiversity. And so one of the end point is that these digital specimens enable us to, one, engage with a wider range of people in biology. You know, people that are interested in phenology. If you're into plants, these digital objects are really useful because you can get phenology data off a botanical sheet, right? It's in flower, for instance. There's possibilities of getting information from morphometrics, automated species identification, things like that. Once we have these digital data and we can share them, you know, this is sort of that old view of museums, right? There's only a few of us in museums to facilitate all that work. Once we have those digital objects, it enables, you know, this universe of people with different interests to actually interact with these objects in ways that are just simply not possible in the physical collection itself, right? We can't fit all of you in the herpetology collection at UF on a single day, but we can do that online. And that's really an interesting opportunity for us to expand the impact of these museum collections. I'll just say, please publish and share your data. If you don't, someone's going to publish data just like it. So you might as well begin getting in the practice of sharing data if you're not currently doing that. And last, I just say that all of this is simply possible because of the diversity of people that we're lucky to have in my lab and at the University of Florida and our network of collaborators. And if we have a few moments, I'm happy to answer questions about the project.